All right, hi everyone. Welcome to the first lecture video. We'll see how this holds up, see if technology works with us and all of that, see if it's useful to you all. I just want to make as many options as possible so that this whole messy situation can go as smoothly as we can hope for. So let me know if this is useful for you, if you have any requests or recommendations, and we'll see what kind of adjustments we can make. So. Right now, I am recording this on March 26th, which is Thursday. And so I'm going to cover the lecture slides from both March 23rd, which was Monday, and March 25th, which is yesterday, Wednesday. Those lecture slides are up on Moodle for you. Hopefully you take a look at those. I know there wasn't much, especially in Monday's slides. Just getting everything together and getting it all reorganized has been a challenge. So I did give you those YouTube links in there that have good information, and of course the textbooks in there as well. So if you have any questions, of course, always reach out to me. But a couple of the housekeeping notes that are current as of today, 3/26, that I'm recording this. So the weekly quiz number 6 opened up today, and we've extended the due date on that since this video and the slides themselves were delayed. So that'll be due on Tuesday, March 31st at 11:59 p.m., so at the end of the day there. We also have the attendance quiz for this lecture, March 25th. That one also opened up today, and so usually I think there's 24 hours to complete the quiz. This time we'll leave it open through the end of the day, Saturday, March 28th. And then there's a couple of things to help you try to stay organized in this whole new format. I posted an example of what the weekly schedule will look like up at the top of our Moodle page. So it can help you keep track of the assignments and everything that you should be keeping up with each day, the quizzes and when the lecture slides come out. And then also on Moodle, all the assignments that I've set up, all those quizzes, they have open and close dates and expected due dates and things like that. So you can refer to the excuse me, Moodle calendar. That should be up to date and easy to follow. And I think you can even set Moodle to set your reminders when things are due or when things open up. So let me know if you have any questions about that, but it's just trying to give you as many resources to keep everything in order as you would need. So, all right, so let's see. Today, we're gonna start with the slides from Monday, talking about pain modulation and the chemical senses. Let's see the housekeeping back there. Oh yeah, so first, I wanted to go ahead and go through the beginning of these slides from Monday, so that if you haven't looked at those, then we can be sure that we're all on the same page for the new format of the class here. So, first thing, most importantly, just know that this is a challenging situation for everyone. And the university and the psych department and I are all aware of that. But we're committed to making that all go as smoothly for you all as possible. So we got about 5 weeks left of the semester here, and we're all prepared to do whatever we need to do to help you all adjust and get through this successfully. So if there's anything that you feel like you need that you don't have already, then let us know. Contact anyone in the department that is appropriate, or myself, and we'll see what kind of adjustments we can make. The schedule and the syllabus may change as needed, so please stay flexible on that, and I'll keep you up to date as any of those changes arise. Obviously, that's exactly what has happened in this past week, so I appreciate y'all's patience and support through that, and I think we've got a system that'll work out now, and I'm getting my head wrapped around everything, and so I think it'll be much smoother sailing from here on out. But just stay flexible, and yeah. So yeah, the most important thing really is take care of yourselves and those around you. So I hope you all are healthy right now, and I hope you continue to stay healthy. And let me know if that changes, and if we need to make any adjustments to accommodate that, we definitely will. So the 4 kind of important points as we move into this new era for these last 5 weeks. Number 1, communication will be key, and especially through email. So check your NC State emails regularly. You've seen that that's been important in the past week as I've had to make changes. So Definitely check that regularly because that's the best way for me to contact you and for you to contact me. Just in case people start having internet issues or computer issues, I know that can happen. So, but I also know that our cell phones and cell service are pretty reliable. So if you need to, you can call or text me. I have my number there. And you can contact me for any reason. So just reach out, anything you need, and we can chat. As far as most like meetings that you would need to set up, we would do that over Zoom or Google Hangouts or something like that online for the most part. So obviously, you know by now that the class is now a completely online format. I've taught an online class before for a few semesters, and so when I put together these slides, I felt pretty prepared for that. But as you've seen in the past week, this has been a totally different animal, so we're making adjustments, but I do at least feel confident that will still be an effective way to learn the material. So one of the most important things I learned from teaching an online class is that technology issues are inevitable. And so to be able to handle those, one, we need to make sure that we don't put off assignments to the last minute because there's very little that I'll be able to do to fix technology issues, especially at the last minute. And so you have to be able to have some time to deal with it and to let me know that you're experiencing technical issues for me to be able to either help you solve them or to make allowances and adjustments as needed. So really, as those things come up, let me know as soon as possible. Third, the grading structure. So all the things that make up your final grade, the weekly quizzes, the exams, and attendance, those are all the same, but each one of those looks a little bit different in the online version of the class, and I'll go over that in some more detail. And then finally, the NC State Bookstore has made their online textbooks available for free through the end of May. And really think you all should take advantage of this. So one, you don't have to carry around that CalIT textbook if you don't want to. You can just have it digitally. And then also these other two textbooks are good resources that I think will help kind of fill in the gaps where we're not very happy with our current textbook. So those are good resources and it's all free. So I do encourage you to go sign up for that. So the main changes to the course right now are that obviously we will not have required class meeting times. Instead, then I have these attendance quizzes that you've probably seen on Moodle already. Hopefully you've already completed those. So basically, from the time that the lecture slides come available, you'll have until midnight on the following day to review the lecture and the lecture video or slides, whatever you're using, and then complete one of those attendance quizzes. So those are on Moodle and they open and close automatically, and they're really easy. It's just most of it isn't actually about the material itself. It's just kind of simple little questions, 4 or 5 questions to make sure that you looked at the slides or the lecture video. Yeah, so those will open and close automatically. So if you miss that deadline, you won't be able to take that quiz. So make sure you get in there on time. The second big change is that instead of our regular required class meeting times, and this is what we have not implemented this week, but we will definitely start it next week. We'll have optional Zoom meeting times during our regular class times. So that's Monday, Wednesday, 3 o'clock to 4:15. And remember, that's Eastern time, just in case you've had to go to another time zone by now. And so during those Zoom meetings right now, the plan is that I will play the lecture video that I record and play that over the live stream and just be available that as you all watch it, then you can ask me questions and I'll pause it and take a break to answer the questions. I might, we'll see how this all plays out and what you all find most useful, but I might, if I have time and everything, still do those lecture recordings and lecture videos ahead of time. But then during the Zoom meeting, I might just do a live stream, like I'll redo the lecture in real time. We'll see if I can pull that off or not. And also you all let me know what would be most useful to you. So I don't think many people are going to take advantage of this option, the optional Zoom meeting, in which case I might cancel them as regular things, but then I'll totally be available at those times for a one-on-one meeting by appointment or lots of other times too. So the Zoom meeting link is up there, and let me know how useful that is to you all and anything else that you would like to see. And then the last big change is to the exams. So those are obviously going to be online. We'll have them on Moodle. I've added a little bit of extra time to each one of those, so instead of an hour 15 minutes, you'll have an hour and a half to complete those. And when you start them, then Moodle will have a little timer on the screen, so it'll show you how much time you have left. After that time, it'll just submit the answers that you've completed if you're not done with it, and it'll close you out of it. I'm not sure if I'm going to make these open book, open notes yet. I feel like I'll probably have to, considering there's no way to really proctor the exams. But so if that's the case, then the questions will be a little more conceptual and kind of difficult. But then you'll have the advantage of them being open book, open notes, of course. And actually, if I go that route, now that I'm thinking about it, I might also at least extend the time that you have to complete it. I'm not sure. Let me think about that, and if y'all have opinions on that, then reach out. Let me know what would work for you. So exam number 3 has been moved. It's now on April 13th, and it'll be available on Moodle for 24 hours. But keep in mind that once you start it, then you'll only have that hour 30 minutes to complete it. And then exam number 4 and the optional cumulative final will be open from April 22nd all the way to the end of the finals period, which is May 5th. Here's a breakdown of our new schedule. So starting from the extended spring break. So here we are in this week, March 23rd and March 25th. We've been going over pain modulation, chemical senses, and movement. And then next week we'll do some more on movement and then move into motivation and emotion. And after that we'll cover cognition. And then on that Wednesday, then we'll have the review session for exam number 3. And I believe we'll use Kahoot again. And I think that we may not do a live version of the Kahoot. I still haven't pinned this down yet. But if not, then I know that Kahoot has an option where you all don't have to do it in real time as a group. You can do it on your own time. But let me think about that, and I'll let you all know as I make a decision. So then exam number 3 is coming up on April 13th. And it'll cover all of those topics, obviously, all the way back to extended spring break. Then after we get back from exam number 3, we have just 2 more full class periods. I guess technically 3 if we include this review session, but the plan is to go over abnormal psychology, drugs, and therapies. And so there's not much material there, obviously. And then we have our review session. So just expect exam number 4 to be relatively easy. It's just covering these 3 topics, and then maybe have a handful of things coming from the previous units as well, but there definitely won't be too much of that in there. For the review session number 4, since there's so few topics in Unit 4, then I will definitely focus on those abnormal drugs and therapy, but then I'll also cover a handful of things from the whole semester so that people who want to take the optional final cumulative exam will have some fresh material that to review there as well. And then on that same day, 4/22, the exam number 4 and the optional cumulative final will both open, and then those again will close on May the 5th. And then by May 7th, then final grades will be posted. All right, so as a refresher, what we went over so long ago now, we were talking about pain sensation and pain perception. And remember that pain is received by specific receptors, we call nociceptors, but they can receive all kinds of different information that is then perceived as pain. So usually these nociceptors are called free nerve endings. They're unmyelinated axons near the epidermis, and they mainly involve the neurotransmitter substance P. And remember that they can receive information including mechanical information. So they have there are mechanically gated ion channels on some of these free nerve endings that can detect pressure, and especially like very sharp pressure, like if you were to be pinched. So they can receive that kind of information. Some of them are sensitive to thermal information, so extreme temperature, like heat. So if you're burned, then some of these free nerve endings have temperature-gated ion channels, so that when the skin gets to a certain temperature, it heats up, like from a match, those ion channels will open, and it'll cause the receptor to fire. And then finally, chemical. And so we talked about this as being good for detecting physical injury to nearby cells. So when nearby cells are injured, they may be broken open and all of their contents flow out into the extracellular fluid. And some of the free nerve endings can have receptors for some of these chemicals that are supposed to be inside the cell, but then end up outside the cell. Things like cytokines, ions, and neurotransmitters. So in that way, then these free nerve endings can detect when other cells are injured nearby. And so they will send a pain signal in response to let you know that you have suffered physical injury. And we talked about the pathway of these pain signals. So they go from those receptors, nociceptors. They send a pain signal up into the spinal cord, and they synapse with an interneuron that will then send that pain signal all the way up to the brain, and specifically to the thalamus. Remember, the thalamus is the sensory relay signal, so that's where most of our sensory signals are sent before the thalamus sends them to whatever part of the brain they're supposed to go to. So in the case of pain signals, some of these are sent to the somatosensory cortex, which, remember, that's where our touch signals are sent, and that allows us to perceive where the pain is coming from. So if you get a pain signal sent from your finger, it goes up to the thalamus and then to the somatosensory cortex. The somatosensory cortex will be able to know that, one, you sense pain, two, it came from your finger. And then some of those pain signals, instead of going to the somatosensory cortex, go to the limbic system, because it'll be, the limbic system, remember, is about processing emotions and memories. And so pain is really important to use in building memories, because you want to avoid pain. So you want to be able to build a memory of what happened when you received pain, so that next time you can avoid that situation. And then we talked about how substance P is the main neurotransmitter used in pain perception or sensation. And that is true, but there is also a role for a neurotransmitter called glutamate. So substance P is most associated for signals of intense pain, and glutamate is most associated with signals for mild pain. So the place that these are usually used is right here, as the pain receptor axon enters the spinal cord and then synapses with this interneuron. It's this gap here where substance P or glutamate is being used to send that signal between the two neurons. And this is what it looks like up close here. So the pain signal is coming in here through this pain receptor axon, and then it synapses here with the interneuron and sends the signal up to the thalamus. So right here I'm showing substance P here, but remember, this could also, for mild pain, be glutamate instead of substance P. And this, as we talked about pain modulation, this synapse here between the pain receptor axons and interneurons in the spinal cord, this will be really important. We'll come back to this. So moving on from, we've already talked about pain, now we're going to talk about pain modulation. And at least as described in the Callett book, the main theory of pain modulation is called gate theory. And so we have these inhibitory signals that come from the brain or from touch receptors. And what they do is they inhibit that the ability of the pain receptors to send their pain signal to the rest of the nervous system. And so by inhibiting that ability to send the signal to the rest of the nervous system, we imagine that as basically closing the gate on those pain signals. And so that's why it's called the gate theory of pain modulation. And there are two different types of inhibitory signals that can be used. One is called the descending. Well, it uses the descending pathway of pain modulation, and it's descending because it's inhibitory signals that come down from the brain into the spinal cord. But then we also have an ascending pathway, and in this case, those inhibitory signals mainly come from other touch receptors. So they're coming from basically the same area where the pain signals are coming from, but they're just simple touch signals instead. So looking at that descending pathway, these inhibitory signals start in the periaqueductal gray area, which is up here in the tegmentum, and it sends these inhibitory pain signals through all the way down into the spinal cord, and it's mostly using neurons that communicate with endorphins. So those come all the way down. I believe I have, I didn't show it. Okay. If you remember this slide, so this is where the pain signals are synapsing with the interneuron. So where this descending pain or inhibitory signal is coming down, it's going to interrupt. This is where it's closing the gate right here. So instead of the pain signal coming from the pain receptor into the interneuron, these inhibitory signals come here and stop this transfer of substance P right here. So you can kind of see that in this image too. So the signals start up here in the periaqueductal gray area, come down through the spinal cord, and then they synapse onto that pain receptor axon. So usually that pain receptor axon is trying to send substance P across the synapse here, but this inhibitory stops this pain receptor axon from being able to send out that substance P. And then the ascending pathway, basically if you remember where this pain signal comes into the spinal cord, it's coming in through a pain receptor axon, but there's also touch receptor axons coming in at that same area. And so the pain signal is trying to be sent from the pain receptor axon across to the interneuron and up to the brain. But what happens is that at the same time, a touch signal will come in, and that touch signal will work similar to how the descending pathway works. Basically, the touch signal will be sent out here, and this touch receptor axon will actually synapse onto the pain receptor axon. And just like the descending pathway, it'll inhibit this pain receptor axon's ability to send that pain signal to the interneuron. So this is, again, where it's closing the gate, it's stopping that pain receptor from sending out the substance P or the glutamate. And so this is something you've probably seen in your own life. Like if you get hurt, maybe if you fall and skin your knee, you get pain in that area. And somehow, if you figured out, if you kind of rub that area, a lot of times the pain will kind of go away. And that's through this ascending pathway, as you touch around that area, touch signals here, synapse onto that pain receptor, and it'll inhibit that pain signal. So that's why you rub a spot when you get hurt there. All right, so we're moving into the chemical senses now, and that's going to be gustation, which is better known as taste, and olfaction, which is better known as smell. So these are chemical senses, and remember, we always, in all the different senses, we want to look at the physical information in the environment that is being received by the nervous system. Because that's what sensation is, right? It's all about taking information from the environment and bring it into the nervous system. So it's chemical molecules, in this case, is the physical information, and those are received into the nervous system through chemoreceptors. So sensory receptors that receive chemicals. And each one of them has, usually it's all about having different specialized receptors on these sensory cells that will allow specific molecules to bind to them. And so if we've talked about similar kind of cells when we talked about receiving the chemical signals in a neuron. So in the postsynaptic neurons, dendrites, remember at the synapse, it has special ligand-gated channels where neurotransmitters will bind to those channels and then open up and they'll let ions in. So in a similar way, these chemoreceptors will allow specific molecules to bind to them, and then something happens. We'll talk about that in more depth later. So the functions of these, of taste and smell, are all about detecting harmful or beneficial chemicals in the environment and in our food. So for example, poison. Maybe you taste some food and it tastes bitter, and so you know that it has, well, usually in the evolutionary sense, it would have some sort of poison in it, so you know not to eat that. And then it can also help us detect chemicals in our environment and our food that tell us about the presence of other harmful and beneficial things. So, for example, maybe you smell ripe fruit around, and that tells you that it can help you find food that is high in energy. I encourage you to watch this video from the Crash Course YouTube series on taste and smell, and it'll go through everything in more depth than I was able to include in these slides. So looking first at gustation, or taste. So remember, the physical information for both these senses is going to be chemical molecules. But in this case, it's chemical molecules that are dissolved in some kind of liquid, and we call them tastants. So the structures here, obviously, taste comes from your tongue. And there are these bumpy structures on your tongue called papillae. And on those papillae, there are taste buds. And those taste buds are where our taste receptor cells are. And we'll talk about those in more depth in the slides from yesterday, from March 25th. So I'll just go over just mention them in brief there, and we can talk about them in more detail later. After those tastants are received by the taste receptor cells in the taste buds, then the signals that they receive are communicated up to the insula, which is the location of the primary taste cortex. So you can see in this cutaway up here, the signals come from the tongue, they go up here through the medulla, go up to the thalamus, of course, because everything goes to the thalamus, and then they end up in this area called the insula. And that's our primary taste cortex, or gustatory cortex, and that's where the perception of taste is processed. One interesting thing about taste is, remember, we've talked about contralateral processing in some of the different senses. In the case of taste, then the taste signals that come from each side of your tongue, so the left or right side, actually get processed mostly in that same side of the brain. So taste signals from the left side of your tongue will be processed in the left side of the brain. So instead of contralateral, this is called ipsilateral. Ipsilateral is being on the same side. And we'll talk about the different tastes in more detail in the next set of slides as well, but there are 5 basic tastes: sweet, salty, sour, bitter, and umami. And again, remember, these are useful for detecting harmful and beneficial chemicals in our food. So for example, poisons usually taste bitter, and we tend to like the taste of sweet foods because sugar is a source of energy. And it's really interesting that our taste preferences at any moment will change according to what our body needs at that point in time. So for example, if you're dehydrated, you'll probably be more attracted to salty tasting foods because salt is really important in maintaining the homeostasis of water in your body and kidney function. And overall, so we have these basic tastes, but what we perceive as flavor is actually a combination of all these basic tastes, but then also of information from the olfactory system. So our sense of smell is really important in our sense of flavor. So again, here's a couple of useful short videos, and I encourage you to watch those. They go into more detail about taste. All right, so the second chemical sense is olfaction, or smell. So physical information, we know it's going to be chemical molecules because both the chemical senses are. But in this case, it's volatile chemical molecules that we call odorants. So volatile just means that they are suspended in the air. So that's why you have to breathe in air through your nose to smell things. It's these volatile chemical molecules. So the important structures here are the olfactory epithelium, which is the collection of olfactory cells that line the inside of the nasal cavity. So nasal cavity over here, and the whole area in this nasal cavity is lined in cells that have their olfactory receptor cells. And together, all of those cells are called the olfactory epithelium. So these olfactory cells that receive these volatile chemicals, these odorants, they're in this olfactory epithelium, and they have cilia, these hair-like structures that extend off the tips of them in the nasal cavity. And it's on these cilia that there are the special receptors where the chemical molecules can bind to. So after the chemical molecules, the odorants, bind to the cilia, then it causes these olfactory sensory neurons to fire an action potential. And that signal comes up here into, remember, the olfactory bulb. The olfactory bulb is the first place in the brain where sensory signals enter. And remember that the smell signals don't go to the thalamus. they actually go to kind of areas, a lot of different places, but mainly in the limbic system, which is why we associate it with, emotional memories. But there is a place in the cortex where some of this smell information is processed, the olfactory cortex, which is in these areas called the piriform cortex and the entorhinal cortex. So some useful functions of the sense of smell, again, detecting harmful or beneficial chemicals in the environment, like poisonous gases, but then also detecting chemicals in the environment that tell us about the presence of other things in the environment that might be good or bad. So you could smell something dying nearby, which means if something died, then something dangerous might have happened or be nearby as well. So you may want to avoid that area. Or if you're a social creature, maybe go help whatever is dying. Or so it can tell you about useful information that isn't actually associated with the smell itself, but it can tell you something about something else in the environment. And then also, again, remember the smell of ripe fruit tells us that those fruit have lots of sugar, which is good for energy. So remember, it combines with gustation or taste to affect our perception of flavors. And it's tied directly to the limbic system here. So smell helps us form important emotional memories. For instance, if you get sick while drinking sour milk, then that memory will be encoded really strongly in your by the limbic system. So your memory of drinking the sour milk and the smell of the sour milk. So that next time we smell sour milk, we'll experience the emotion of disgust and avoid it so that we don't get sick. And we'll talk about disgust when we talk about emotions and motivation, but these are really powerful motivators, emotions are, to remember when you did something good or when you did something bad. All right, so that concludes the slides from Monday, which was March 23rd. So now we'll move into the slides from yesterday, March 25th, going over chemical senses in a little more detail, especially gustation, and then getting into movement control a little bit. Here's a picture. This is in the textbook. Here, this is the axon of a motor neuron. And you can see that it has several different branches here connecting to, each one is connecting to an individual muscle fiber. We'll talk about that in these slides, but just important to see that one axon of one motor neuron can connect to a bunch of different muscle fibers. But first, let's see, we've already talked about these housekeeping notes. Here's just the chapter in Calit for talking about the control of movement. So yeah, first we're going to go into some more detail about gustation. So look at the structures again. The tongue, pretty easy. I think everyone knows the tongue. On the surface of the tongue are all these bumps and ridges of tissue called papillae. And you can actually see these with the naked eye. A lot of people think, at least when they're growing up, they think that those bumps are the taste buds, but the bumps that you can see are actually the papillae. So I think there's 4 kinds of papillae total, but we'll just talk about these 3 kinds because they're the ones that hold the taste buds. The other kinds of papillae don't have taste buds on them. But each papillae itself will have at least 10 taste buds, and we have 3 different kinds called the circumvallate papilla, or papillae is plural, but circumvallata papilla right here. Here's a foliate papilla and fungiform papilla. And remember, on these, kind of lining some of the sections of these papillae are taste buds. So taste buds are the structures in the papillae that hold the taste receptor cells. So they're these kind of barrel-looking structures here. And so some of these cells are just structural cells, but some of them are taste receptor cells. And each taste bud will have about 50 of these taste receptor cells in it. So remember, gustation, olfaction as well, are both about receiving chemical information in the environment, little molecules. And gustation is, we call those molecules tastins, ones that dissolve in liquid and then can be binded to these taste receptor cells. So these are the 5 basic tastes that we talked about. bitter, sweet, sour, excuse me, umami, and salty. So here's a couple of depictions of taste receptor cells. And the big difference between them is that one type of taste receptor cell has these ligand-gated G protein coupled receptors. So we've talked about these before when we talked about metabotropic effects. You can see that in the earlier slides from one of the other units if you want to refresh on that. Basically, it's these receptors that stretch across the cell membrane, and they have a special site on the outside where molecules can bind to. And when that happens, then inside the cell, this little thing called a G protein will break off from the receptor, and it'll float around and it'll bind to other molecules inside the receptor cell. One common one is this cyclic AMP, the CAMP. So that's just a molecule that floats around the inside of some cells, and when a G protein breaks off and binds to it, then it basically turns into an energy carrying molecule so that then it can go tell other things to happen in the cell, like maybe start transcribing certain genes, or it'll tell other molecules to come into this area and things like that. It's got a wide range of effects that can happen. Remember those metabotropic effects. So anyway, the types of taste that are associated with these G protein coupled receptors are these three: sweet, bitter, and umami. So sweet, the molecules that will bind here are sugars, obviously, and then some other molecules like some proteins and stuff, and artificial sweeteners, those kinds of things will bind to these same receptor cells. And so these, again, do metabotropic effects of various kinds, and in general, the main function, kind of evolutionary function of being able to taste sweet things is detecting that these foods are rich in energy. Then kind of at the opposite end of that spectrum, so we definitely were attracted to sweet foods. The other end of the spectrum would be bitter foods, because their function is for detecting toxic chemicals for the most part. So in general, if something tastes bitter, we don't like the taste of it, and it's because evolutionarily that bitter taste was associated with chemicals that were bad for us. There's a lot of different chemicals here. There's a lot of different types of taste receptors that can detect bitter tastes, but they have the same effect or they work in the same way as sweet tastants do, where they bind to this G protein coupled receptor and cause other things to happen in the cell. So the last basic taste that works with these types of taste receptors is umami. It's kind of like, I think they describe it as kind of a meaty taste. You can taste it in meats and cheese and stuff. So the molecule that would bind to taste receptor cells in this case is called L-glutamate, and it's useful for detecting protein when you're eating. So if your body feels like it needs protein to build muscles or anything else, then you might crave umami flavors. Then over here, this other type of taste receptor cell is much simpler. It's basically just got, instead of a gated ion channel or a G protein coupled receptor, excuse me, they just have these ion leak channels. So they're just protein channels that are totally open, so ions can move in and out of them freely. Other molecules can't, because ions are small and simply shaped, so they can move freely, but more complicated molecules like proteins wouldn't be able to fit through here. So it allows the passage of ions to move in and out. And for sour, the ions that are associated with it are hydrogen ions. So just a single hydrogen plus here, also called protons sometimes. But basically, if there are a lot of hydrogen ions around, that's how you define if something is acidic or not, is the number of those protons, free protons. So if there are a bunch of those around, they'll float into the cell here. And remember, they're positively charged. So as they float into the cell, then the cell, which is usually at negative 70 millivolts, remember, that's the resting potential. As you add positive charge in there, that'll start to depolarize the cell, and that'll cause the cell to fire, just like any other time that we've seen positive ions moving into a cell. So in this case, when positive ions like hydrogen move into these types of cells, these taste receptor cells, then they'll fire, they'll send a signal, and telling the nervous system that it's detected something sour. There's some similar taste receptor cells that again have these ion leak channels, but they'll mostly allow other positive ions besides hydrogen. So mostly sodium, but then also some other ones like potassium and lithium. They can move in and out of the cell, and it'll work the exact same way as the sour receptor cells work. So as these positive ions move into the cell, then it'll cause the cell to depolarize, and just like we saw in regular neurons, voltage-gated ion channels will open, and then that'll cause neurotransmitters to be released and send the signal of salty. So detecting salty or salt in our foods is useful because, remember I said that it's useful for maintaining water homeostasis. So like basically the amount of water that is either in your blood vessels or in the extracellular fluid in your body. And a lot of this is determined by not determined, is handled in the kidneys. And that's where salt plays a role. So that's why it's important to detect salt. All right, so that was the last additional bit of info about the chemical senses. But I did want to talk about this idea of signal strength, because a lot of times we'll talk about how neurons can either send a weak signal or a strong signal. So I want to go over what that really means, because remember, when neurons send a signal, they fire an action potential. Action potentials are always, they look exactly the same every time they're fired. So they are always the same height, and they always take the same amount of time to depolarize and then repolarize and then come back to resting potential. So one action potential always looks the same. You can't make that bigger or smaller to change the strength of your signal. Instead, the neurons, if they want to send a stronger signal, they can do that in ways that relate back to summation. So remember, summation is when graded potentials are added together, their effects are added together in the postsynaptic neuron to determine if the neuron should fire or not. So in the idea of spatial summation, if remember when we talked about summation before, it was all about just one neuron communicating with one other neuron. And or I'm sorry, in spatial summation, we talked about it being useful for having multiple neurons talking to one neuron. So, but in this case, one single neuron can also use spatial summation to send a stronger signal. So the way that this would work is that if one neuron wants to send a stronger signal over here to this neuron, then instead of just being attached like this one at one point here, it'll increase the number of connections that it has. So that way, whenever it fires once, it'll actually release neurotransmitters at several different places and cause graded potentials to happen in this postsynaptic neuron at several different locations. So this actually takes a lot of energy and resources and time. So if a neuron feels like it needs to send a stronger signal on a regular basis to another neuron, it would use this kind of spatial summation approach. It would grow out more axons or branches of its axon and synapse to more dendritic spines on the postsynaptic neuron. But if a neuron needs to send one signal that's really strong right now without having to take the time to grow off new branches and stuff, it can do that through a process that's similar to temporal summation. So instead of sending signals to several different locations on the postsynaptic neuron, it'll just send a bunch of signals all at once, or kind of one after another really quickly to the same synapse here. Kind of looking at this, if this is a neuron that wants to send a strong signal, and it'll do that through temporal summation. And this is a neuron that wants to send a strong signal, and it'll do that through spatial summation. So this one just basically has one connection here to the postsynaptic neuron, and this one has several connections. So each one fires up here, so it's sending a signal. That'll move down the axon. And then on this one that is practicing temporal summation, as this action potential is moving towards the postsynaptic neuron, as soon as it can, then this neuron will fire again and start sending another action potential down its axon. In reality, it probably wouldn't happen while this action potential is still traveling down the axon. It would probably have to wait until this action potential reaches this neuron. But just for simplicity's sake that I'll show a whole train of them happening. Over here, there's still just this neuron just fired once, remember? The difference is for this one that is undergoing spatial summation, it's got several branches here. And so that one action potential will actually travel down 3 different branches of the axon and kind of act as 3 different action potentials. So now each one of these branches is sending that action potential signal. Meanwhile, over here, that temporal summation neuron just sends, remember, several one after another action potentials all in really quick succession. So the end result here is that in the end, this postsynaptic neuron got essentially three action potentials from one neuron that all kind of got here at the same time coming through different synapses. And this one is also going to have 3 action potentials reach this postsynaptic neuron. It just had to fire an action potential 3 different times over and over again really quickly. All right, so now coming into the control of movement. Remember, this is going to be in CALIT 8.1. And we'll just go over a few things now, and then we'll go into this in more detail on Monday. So starting off, movement, of course, is handled by our muscles. So muscles are these basically bundles of these structures called muscle fibers. And these muscle fibers are actually 1 cell each, but they're really long and stringy. So this would be 1 muscle fiber here, which is actually 1 cell, but it's really big. And so muscles are made up of whole bundles of these muscle fibers. And each muscle fiber can either be what's called a fast twitch fiber or a slow twitch fiber. We'll talk about that in more detail in a bit. But when a muscle is made up of all these muscle fibers, it could be made up of a whole mixture of fast twitch fibers and slow twitch fibers. So different muscles will have different combinations of these, and different people will have different combinations of these too. So each muscle fiber is attached to 1 motor neuron. So you can see this is the axon of a motor neuron here, and it's attached to several different muscle fibers. So whenever this motor neuron fires, what it's basically doing is it sends this motor signal to these muscle fibers, and it'll cause these muscle fibers to contract. So if all of the muscle fibers in this muscle receive a signal to contract, that whole muscle will be contracting and causing whatever movement to happen that related to whatever that muscle is attached to. But importantly, the fewer muscle fibers that each motor neuron is attached to, the more precise movement will be able to happen from that muscle. So in the book, they give the example that in the eyeball, 1 motor neuron might only attach to like 3 different muscle fibers, whereas in the bicep, 1 motor neuron might attach to like 100 or hundreds of different muscle fibers. And obviously, you can have much more precise movement of your eyeballs than you can your bicep, and that's because there's more motor neurons available to control the movement in the eyeball. And where the muscle or the motor neurons attach to the muscle fibers, we call those neuromuscular junctions. So it's basically just a synapse, because the motor neuron is sending a neural signal to these muscles, but it's a synapse between a motor neuron and a muscle fiber. So a little more detail about the structures here of muscles. Remember, I said that there's slow twitch fibers and fast twitch fibers. And slow twitch fibers are they undergo a process of using and creating energy that we call aerobic, an aerobic process. So it uses oxygen and glucose to produce the energy that it needs, an ATP, to produce those movements. So these slow twitch fibers produce really slow movements, and they don't fatigue very easily. So these are a muscle that is high in slow twitch fibers would be useful in like marathon running. So people who are long distance runners probably have a lot of slow twitch. They can keep moving over and over again for a really long time. And then The other type of fiber is called fast twitch fibers, and these are anaerobic. So these don't use oxygen to produce the energy that they need. They just use glucose in an anaerobic process, and so it doesn't create as much energy, which is why these fast twitch fibers would fatigue more easily. Basically, they run out of energy because using oxygen allows you to create more energy. So that's why these slow twitch fibers can last longer. But without oxygen, then you just don't have as much energy to keep going. But the advantage here is that you can't produce as much energy, which is what ATP is. Remember, it's one of the energy holding molecules, but you can produce energy more quickly. And so the anaerobic fast twitch fibers are good at making really fast movements. They just don't last as long. And then one kind of classification of muscles that we have are antagonistic muscles. So these are muscles, sets of muscles that work in opposition of one another to move a limb in different directions. So when one of the muscles contracts, the other one will relax so that that limb can move in whatever direction the other muscle is contracting. So when we look at this, for instance, here's a leg, and you have on one side of the knee joint, you have the hamstring, and on the other side, you have the quadricep. So the hamstring, so these are antagonistic muscles to one another because one causes the knee to move one direction and the other one causes the knee to move the other direction. And so in this case, the hamstring is called a flexor because it causes the limb to bend. And in this case, the quadricep, the other muscle in this antagonistic group, is called an extensor because it causes the limb to straighten out. So another example of antagonistic muscles would be the bicep and the tricep. And in this case, the bicep would be the flexor because it causes your arm to bend, and the tricep would be the extensor because it causes your arm to go straight. And there are three general muscle types: smooth muscle, striated or skeletal muscle, and cardiac muscle. So the smooth muscle, remember, they all consist of muscle fibers, but in smooth muscle, they're these long, thin fibers. And you mostly find smooth muscle in the digestive system, controlling things like your intestines and other organs. Striated or skeletal muscles are the ones that are involved in the control of movement of your body parts. So, moving your arms and legs around. These are long cylindrical fibers, and they have this kind of striped appearance to them. Striated basically means striped, so that's how you can remember that. And motor neurons communicate with these striated skeletal muscles with acetylcholine, like pretty invariably. So when you think about the neurotransmitter that's most associated in sending motor signals to, like directly to the muscles, striated muscles in particular, it's going to be acetylcholine. And then finally, there's cardiac muscle, and these are just found in the heart, obviously cardiac. These consist of fibers that are fused together at various points. So whenever they contract, they all contract together. It's different from these other ones because those muscle fibers aren't attached to one another, so the fibers can actually move independently, but that doesn't happen in the heart. So obviously the function of these is to control the beating of the heart, cardiac muscle. And the question I posed here that y'all can think about is that each one of these types of muscles, smooth, skeletal, and cardiac, is controlled by either the autonomic nervous system or the somatic nervous system. And so think about knowing what you know about each of those systems, which one of them would control each of these types of muscles. So is it the autonomic or somatic nervous system that controls smooth muscle? And which one controls striated or skeletal muscle? Which one controls cardiac muscle? So think about that. And then we've talked about proprioception already. You can go back to the lecture on the mechanical senses when we were talking about touch and all that, because similar mechanical receptors are involved in proprioception. So you can go back to, those slides if you want a refresher. That proprioceptors or proprioception is this sense of where your body is, what the limbs are doing. So muscle control with these proprioceptors allows us to make fluid movements, and by processing our movements in the spinal cord, kind of making fine adjustments without having to get the brain involved. The proprioceptors and our spinal cord can kind of make small adjustments to movements. And so since these are processed in the spinal cord, then not the brain, we call them reflexes. So one of the types of proprioceptors are these muscle spindles we've talked about before. They're in the muscles themselves, and they can detect when the muscle is being stretched. And if a muscle is being stretched, then it'll cause it to contract so that it doesn't get stretched too much. And then in a similar way, there are these proprioceptors called Golgi tendon organs, and they do a similar thing. They act as a brake against excessively vigorous contraction. So in the other direction, instead of being stretched too much, they're contracted too much. And so they'll send an impulse to the spinal cord where whatever motor neuron is causing the muscle to contract, it'll tell that motor neuron to stop sending that signal. So one place that we've seen the muscle spindles play a role in reflexes is in that patellar tendon reflex. So when the doctor hits your knee with the little hammer and it causes your knee to kick, that's handled by muscle spindles, sending a signal into the spinal cord. And one place where, well, this just depicts how a Golgi tendon organ would work. So it detects tension on a tendon, so it knows that this muscle is being contracted, and it wants to stop that contraction from happening. So the Golgi tendon organ will send a signal into the spinal cord. It'll synapse with an interneuron here, and that interneuron will send an inhibitory signal to the motor neuron that is causing that muscle to contract. So it'll stop that contraction. So that's everything that we have for this time. I'm sorry for the delay on this lecture video and that there's not as much material in here as usual, but we will be doing this regularly for starting next week on Monday. And then also we will regularly have those optional Zoom meetings available. So hope this was helpful and that those other resources, the other YouTube videos are helpful. Let me know if there's anything else that you would like to see. And of course, come to those optional Zoom meetings if you want a little more interaction. And I'll probably do live lectures at those. But until then, have a good weekend.